We have all seen a lot of Hollywood movies, and a lot of them have shown scientists giving their experimental drugs to rats. But why? Let's find out in this video why rats are chosen for experimentation.
After injecting the mouse with an IP injection of a ketamine xylazine cocktail according to the text protocol, perform a tail pinch to check the level of sedation.
Let's go.
He's going to test whether these chemicals change the stopwatch mechanism that allows the rats to measure time.
The device attached to this mouse's head can wirelessly inject drugs deep into its brain at the click of a button.
The history of using rats in the lab goes all the way back to the 16th century. At that time, rats were being used for research purposes on their diets. Particularly albino rats were chosen for scientific studies. The reason for choosing albino rats specifically was that they have a similar digestive system as humans, they grow quickly, and many more reasons will be discussed later in this video. It wasn't until the year 1906 that the first rat developed to serve as a model organism at a time when laboratories primarily used the house mouse. The development of the laboratory rat as an experimental model began in the Worcester Institute. In 1906 they developed the Worcester rat model, scientific name Rattus norvegicus. Nowadays, the Worcester rat is commonly used globally as the laboratory rat. There are many reasons for choosing rats for experimentation in labs. But we are only going to discuss some of the important reasons in this video. The main reason being is their genetic, biological, and behavior characteristics closely resemble those of humans. According to a study by Foundation for Biomedical Research, genetically, we humans share nearly 97% of our working DNA with rats. Thus, scientists can mimic in rats the effect of DNA alterations that occur in human diseases, and carefully study the consequences of these DNA misspellings. Rat models also afford the opportunity to test possible therapeutic agents, and evaluate their precise effects. This makes the test to be more precise on rats. Nearly 95% of all lab animals are found to be mice and rats. Biologically, the most important reason for choosing rats is that they reproduce quickly and have a short lifespan of 2 to 3 years, so several generations of mice can be observed in a relatively short period of time. Also, we can test a lot of the human diseases in mice and rats, so that we could research them. Here is a list of some examples of human disorders and diseases for which mice and rats are used as models include. Rats have demonstrated their ability to get addicted to substances, just like humans. They develop personality traits similar to humans, being optimistic or pessimistic. Rats also giggle when tickled and apparently look forward to having sex. These are similar to human characteristics. In addition, like humans, rats learn to avoid unpleasantness and seek rewards. Based on their personalities, they will exhibit varying degrees of tenaciousness to get what they want, or to avoid what they don't want. There are other people against animal testing believe there should be another alternative, but at this moment that's not the case for effective research. As of right now, the FBR states that there is no comprehensive substitute for animal testing and research. Though computer models, cell cultures as well as other research methods does reduce the number of animals used, there is no way to completely replace animal testing, because the pathway to fully duplicating a whole living system does not yet exist. Cell cultures and computers are limited in what they can model.